Hello， 好，感谢大家今天的参与我们今天的研讨会。 那今天的研讨会的主题呢，主要是要跟大家分享一些比较新的、特殊的免疫荧光染色的一些方案跟技术上的更新。那在免疫荧光染色当中，我们可能会有一些问题，包含了一些就是可能会有物种上面的 cross 交叉上的反应，或者是一些背光上的问题，还有一些可能会有一些穿透率或者需要高显析度的相关的试剂。那在这边的话呢，就是很荣幸邀请到我们的讲者 Dr. Klaus Herick。那这边的话就是会跟大家做一些不同应用上的分析。那 So thank you for your attending our seminar to learn about recent innovations and advances in reagents used for immunoforensics microscopy. And we are pleased to invite our Dr. Klaus Herick share this information, including novel antibody labeling and advanced immunoforensics tests and nanobody-based reagent for various immunoforensics applications. And so please feel free and join with us. Thank you.
Thank you, Siri, for your kind introduction. My name is Klaus Herick. I'm based in Germany, and I look after the APAC region for Proteintech. I will talk about innovations and advances in immunofluorescence, and I hope everybody can hear me well. Also, my colleague at the computer for those that are watching us online. Content of my presentation, I will give a quick introduction and then introduce multiplexing, talking about Coralite plus dyes, antibody labeling technologies, a novel antibody labeling kit called FlexAble, our Coralite conjugated antibody portfolio, and then swap into nanobody-based reagents for immunofluorescence. And here I will talk about secondary nanobodies, so-called secondary reagents, conjugated nanobodies, as well as intracellular nanobodies for live cell imaging. Quick introduction into Proteintech. Proteintech comprises 3 brands. There is the Proteintech brand, which is for all of our antibodies and immunoassays, Chomotech for all of our nanobody-based reagents, as well as Humankind for our GMP-grade cytokines and growth factors. Here in Taiwan, we are distributing exclusively through AppReal, and I think if you click on this QR code, you get straight into my colleagues at AppReal. So, what's the Proteintech mission? Our mission is to produce in-house antibodies for every single protein coded for in the human genome. And so far, we got 2/3 of the human genome covered. That is antibodies against more than 11,000 different targets. That's incredibly broad target coverage. So we are ready with the antibodies of tomorrow. So you may identify a protein of interest that you want to study. We may have the antibody for that. And up to now, we have more than 190,000 citations, turning 200,000 by the end of the month, we expect. So antibodies, quick introduction into antibodies. One significant problem of antibody sourcing is that there are a lot of manufacturers that sell to even more resellers, and they may rebrand those antibodies and they sell on to the user. So as a user, you may buy 5 different antibodies, but they may all come from one manufacturer, and you spend a lot of money for multiple times the same product. At Proteintech, we don't do this. So our products are in-house manufactured. We don't OEM. We always provide our original validation data, and we sell from Proteintech, as we call, from our bench to your bench. That's very important for us. In terms of antibody types, we have polyclonal antibodies as well as monoclonal antibodies as well as recombinant antibodies. You may consider polyclonal antibodies being very old and old-fashioned because it's technology out of the 1970s, where I expect none of you but me have been around. Polyclonal antibodies still have lots of advantages because they are an IgG mix. They cover multiple epitopes, so they are really good and ideal for low abundant targets. If you look into cross-link fixed samples, also for rare species due to some cross-reactivities, and into mutated proteins. So also they are highly sensitive and have a good sensitivity. They have obviously some disadvantages, which is a lot-to-lot variability in the background. The monoclonals are detecting a single epitope, better for higher abundant targets. They have, because it's a single epitope, a lower background, but it's a poorer epitope accessibility and less applicability on mouse-on-mouse detection. Recombinants are really good for long-term projects because of the reproducibility, but the target range is very, very limited. I think we have a few hundred compared to 15,000 in total. You can see the problem of it. So all three types of antibodies have their advantages and disadvantages. Multiplexing, I would quickly like to introduce what's multiplexing and then talk about the difficulties. Well, multiplex assays, that's the simultaneous detection of several targets in a single sample by using multiple antibodies, and you generate a detailed snapshot in a complex biological system. What are the difficulties? There is two ways to detect or to conduct multiplexing assays. One is the direct approach, where you use an antibody that is a primary antibody that is labeled with fluorophores, or the indirect approach, where you use a primary antibody and detect the primary antibody by applying fluorophore-labeled secondary antibodies. By going indirect means the process is rather lengthy, complicated, it may be expensive. You may also go the direct approach that eliminates the secondary antibody incubation. You can use same species antibodies using this way. It's quicker, it's simpler, and it may save money. And I will now explain different ways of direct labeling and introduce indirect methods by introducing nanobodies. Direct labeling, for instance, is our coralide conjugated primary antibodies, where we have already more than 2,000 products. And these 2,000 products cover more than 1,000 targets. So on average, we have about 2 colors per target. The products span all major targets in neuroscience, stem cell, and cancer. Coralide and Coralide Plus are very bright fluorescent dyes based on those chemicals that have been covered under the Alexa fluor patents that run out of patent protection. We have 4 colors at the moment, and those dyes are also compatible with cyclic immunofluorescence. And here a few examples are shown. Well, if there is no antibody that is labeled around, and you may have to label your particular antibody yourself. And antibody labeling is also comprising some advantages and disadvantages. And currently there is 2 antibody labeling conjugation technologies around. One is called NHS ester chemistry, and the other is called Malamid chemistry. NHS ester chemistry is the covalent conjugation of surface lysines and the N-terminus. So any amino group that is not protonated can react to the NHS activated dye. It is a random conjugation because you cannot predict what lysine residues are actually, or what amino groups are actually, amine groups, are actually conjugated. you may damage the antibody by conjugation of the paratope. It could happen that that the binding domain of the antibody is labeled and hence damaged. It has limited buffer compatibilities. The buffer for in that the reaction is taking place shall not comprise any BSA, any glycerol or amine buffers. So TRIS doesn't work. Antibody is also lost because you have to, generally speaking, to do a buffer exchange, and a high antibody quantity and concentration is generally needed, but also please keep in mind that these activated dyes, when you open the vial, humidity gets into the vial and the dye loses its reactivity. So once you opened an activated dye, you have to consume it. Null-limit chemistry is the covalent conjugation of cysteines. And in order to label those cysteines, you have to reduce them so that they are available as H group. It is site-directed because there are not so many cysteine residues within an antibody. But keep in mind that an antibody may be damaged due to disassembly of the heavy and light chains at the hinge regions of the antibody are held together by cysteine bridges. Also here we have limited buffer compatibility. You need that reduction step prior to labeling. So you may have to do a buffer exchange and may lose antibodies. And again, it's likely that you need a higher antibody quantity and concentration in order to do the labeling. As again, the maleimide activated dyes also, once you open them, they will start to lose their reactivity and you have to consume the vial in a single go. So as you can see, different technologies, but not really that straightforward and ideal. So how could the ideal labeling technology of antibodies look like? Well, it is desirable to have a strong binding of the dye and the antibody. It should not damage or disassemble the antibody. It should be compatible with whatever the antibody is stored in, what buffer it is stored in, and it should not require a high antibody quantity and concentration. So under ideal terms, you want to use the antibody as it is and just label it. We believe a solution for that problem is our FlexAble antibody labeling kit. It's a novel kit that uses affinity linker to label antibodies with fluorophores in any buffer condition and in any antibody concentration. So in a nutshell, what is it? It is compatible with any primary antibody. It works with any antibody concentration and formulation. It labels as little as half a microgram. You don't need to exchange the buffer. It is very fast and easy. It's just 2 times 5 minutes incubation. You can label many different antibodies with a single kit. You don't need any additional equipment, and it's available for rabbit IgG, mouse IgG1, and mouse IgG2A. How does it work? It's very simple. The kit comprises of a so-called flex linker that you can see here, and the flex linker is non-covalently binding with a very, very high affinity, binding tightly to the primary antibody. And the second step, you add a so-called flex quencher that will bind to unbound flex linkers, and you will apply that mixture to your sample, wash the flex quencher away, and that results in a homogeneous, well-defined, and fully functional labeled antibody. You won't have any aggregation of the antibody or any loss of the antibody activity during the labeling, as the flex linker is targeting specifically the Fc domain of the antibody. The flex linker is a small polypeptide with covalently conjugated dyes. There is 2 dyes conjugated to a flex linker, and hence any antibody is labeled with 2 fluorophores. So degree of labeling of a primary antibody is 2. It has a very high affinity to the Fc domain of an antibody, so it binds in a site-specific manner. We have it specific to mouse IgG1, IgG2A, and rabbit IgG. Rat IgG is to come. And what is the key of this kit is its affinity, because the affinity is high enough to avoid linker dissociation. That's very important. And as I mentioned earlier, any linker comprises 2 dyes, so you have a degree of labeling that equals 2. Protocol, really simple. What is important is that first you equilibrate your reagents to room temperature in order to drive the reaction in the manner it's shown here. And then you mix as little as half a microgram of antibody with 1 microliter flex linker in 8 or up to 8 liter flex buffer. You may apply more antibody and linker by multiples of half a microgram and 1 microliter flex linker. Incubate for 5 minutes, add 2 microliters of flex linker or correspondingly more. Incubate for 5 minutes, and then the antibodies are ready to use. And that's really straightforward. You can set it up within 10 minutes in just 2 steps. How to do multiplexing with FlexAble? There are 2 ways to do. So if there are antibodies from different species, like rabbit IgG and mouse IgG1, you can label those 2 antibodies simultaneously with the corresponding flex linkers. Then you add your quencher and apply to your sample. So that's very straightforward, very simple, very fast. You don't have to consider any particular differentiation. Antibodies from the same species. So like shown here, let's say we have 4 different rabbit antibodies. You have to label separately. So the first rabbit antibody with the green one, the second rabbit antibody with the orange flex linker, the third rabbit antibody with the red flex linker, and the 4th rabbit antibody with the magenta. And you quench and then label, mix those antibodies and apply those to your sample. So it's also straightforward, but it takes a little bit of more preparation, as you have to label same species and same subtype antibodies separately. As I mentioned earlier, those antibody species are available, and we have those fluorophores for immunofluorescence, 488, 555, and 647 is applicable. Some microscopes go into the near infrared range already, but generally speaking, these are mainly used in flow cytometry application. A few words on the Coralite fluorescent dyes, which are novel fluorescent dyes with improved photostability and quantum yield. So what is Coralite and Coralite Plus? So a conventional dye conjugation on an antibody is that you conjugate the dye directly to the amino acids, lysine residues on the surface of an antibody. Yeah, you have a rather low degree of labeling. If you increase the degree of labeling, like here, you may run into dye quenching, where the dyes quench the signal themselves. These conjugates are more likely to aggregate because every dye is very hydrophobic and may precipitate the antibody, the labeled antibody. The conjugates may also have more non-specific binding and have a high background. And what's the new thing about this Coralite Plus technology? It's the so-called DPEC linkers, which are distinct polyethylene glycol linkers. Distinct means the lengths of these antibodies are distinct. Other pegylated dyes have a variety of lengths. These DPEG linkers of the coralite are very defined, and that means you keep the dye in a certain distance from the antibody, so there is no quenching dye to dye. There is no quenching with the surface of the antibody. So the dyes are in a rather defined dielectrical environment and have a very bright, very stable fluorescent signal. So you can increase the level of the degree of labeling. You have a low level of dye-to-dye quenching. You have no aggregation because the pegylation improves the solubility, both of the dye and the conjugated antibody, and you have less non-specific binding. A few data, not a lot, just a comparison of here in yellow, in magenta, coralite 647, and compared with regular 647 dye. As you can see, over time, the stability of the coralite is already good. If you compare that with the plus signal, and here we have the coralite plus versus a regular Alexa Fluor 488, here it is, you can see that the average normalized value of the signal intensity over time is really higher. That's the portfolio. It has 4 dyes, 405, 488, 555, 647, and 750. Very conventional dyes that microscope settings are compatible to. And now I like to talk about nanobodies, which are binding domains of heavy chain antibodies. So what's a nanobody? If you consider a traditional antibody, comprising of a heavy and a light chain, size is about 150, 160 kilodaltons. Camelids like alpaca have these traditional antibodies as well as so-called heavy chain antibodies. Heavy chain, as the name says, is deployed of light chains, but not only it does not have the light chains, it also does not have the CH1 domains. So the size of a heavy chain antibody is about 100 kilodaltons. The binding domains of these, what's shown in green here, that's a nanobody. It's about 30 to 15 kilodaltons in size, 2 to 3 nanometers, about the tenth of the size of a traditional antibody. And when you label those nanobodies with fluorophores, we call it a nanobooster or a nanosecondary reagent. I like to introduce nanosecondaries, which are nanobodies against IgGs conjugated with fluorescent dyes. I also like to introduce nanoboosters and labels, which are also nanobodies against protein tags as well as endogenous targets. And I like to introduce intracellular nanobody probes that are basically comprising of plasmids encoding for a nanobody and a fluorescence protein. Starting with the nanosecondary reagents. So what we have here is we have an antibody, a primary antibody that is detected with a mixture of 2 nanobodies. Here in this case, one Fc specific and one Fab specific. All of these antibodies are carrying 2 fluorophores. A total of 4 nanobodies is binding to 1 primary antibodies, and this results in a degree of labeling of 8 fluorophores per primary. So this is a stoichiometric labeling, very defined and very repeatable. The size and the defined binding of the nanobodies to the primary antibody results in a higher image resolution with a high specificity and a low background. It has no cross-reactivity, and they are highly isotope specific. You can incubate the primary and the nanosecondary reagent in one step, so you save time. They are recombinantly manufactured for high lot-to-lot consistency, and they are available for rapid IgG, mouse IgG1, 2b, and mouse IgG3. Mouse IgG2a is in development, and we have validated it for immunofluorescence, including wide field, confocal, and super resolution. So here's some data. So this is a super resolution image of the lamina of the cell using stent technology, and you can see at this resolution you can see the 3 layers of the lamina. Here is some customer data. That's the 3D SIM microscopic technologies, and what you can see here is mouse kidney sections where surface protein of podocyte cells has been labeled, and the customer gained 20 nanometers in resolution. So by using the primary antibody and the conventional secondary antibody, this resolution was about 130 nanometers. By using the same primary antibody with nanosecondaries, he gained 20 nanometers, and his resolution was now 110, just 110 nanometers, which works in the first instant and is quite impressive. Due to the size of the nanobodies or the nanosecondaries, you also get a more homogeneous sample staining. So what you can see here is again customer data out of Canada. It's a Drosophila lava brain, and it is labeled here on the left-hand side with an anti-lamine primary antibody and an anti-mouse IgG nanosecondary reagent. And you can see the red signal with a blue Darby counter stain. The red signal is homogeneously and equally distributed through the entire sample. And here on the right-hand side, you can see an anti-lamine primary that was detected with a donkey anti-mouse secondary antibody, and you can see that here the red signal is mainly on the surface of this lava brain section, as you can see it here. So it's the small size of the nanosecondaries provide a more equal distribution of tissue samples and thick samples. Also, it's important to know that the nanosecondaries have a very, very low background with no cross-reactivity. So what's shown here is a triple mouse primary antibody stain. So there are 3 mouse primary antibody used for the staining. So it was mouse IgG1, 2b, and IgG3. And there is no crosstalk. So it's a very, very good tool for multiplexing. And we have backed up the data with this dot plot, where we have used a series of our nanosecondaries against different IgG samples applied to the membrane. And the mouse IgG1 detects only mouse IgG1, 2b detects only mouse 2b. The nanosecondary against mouse IgG3 detects only mouse IgG3, and the rabbit detects only rabbit. We've compared that with a leading competitor goat anti-rabbit IgG polyclonal antibody that was pre-absorbed, and you can see, of course, strong binding to anti-rabbit, but also some faint binding to other subclass and species that results into background and unspecific labeling. I mentioned it already, you save time by one-step incubation. So a standard workflow with conventional secondary antibodies, you have your paramelized and blocked cells, fixed paramelized and blocked cells. You apply your primary antibody, incubate for an hour, wash, and then incubate the fluorescently labeled secondary antibody for an hour, wash, and then detect. When you use nanosecondaries, you take your fixed paramelized and blocked cells and co-incubate those cells with the primary and the nanosecondary for an hour, wash and detect, and you can save about half of the time of a conventional workflow. The results are the same. Here is sequential staining, here is one-step staining. We've done tons of experiments to prove that this holds true. Now, I like to quickly introduce advantages of nanobodies for immunofluorescence in terms of resolution and tissue penetration, talking about so-called nanoboosters, which are nanobodies against GFP and RFP that stabilize, retain, and enhance the signal of those RFP or GFP proteins. So they boost the signal, and nanolabels are used for staining non-fluorescent peptide tags or endogenous samples. And here we have a histone sample, a spot label, and a limiting label. Again, it's a nanobody that is conjugated to dyes. The boosting is seen as important for applications where you have to reactivate and enhance this fluorescent signal after bleaching or harsh treatments like for fish or click chemistry that you may have applied to your sample. And here some data are shown for RFP signal after bleach. So here's 100% signal intensity after bleach. And when you apply the RFP booster, you can see that after bleaching, the signal is restored. And here we have an increase of the GFP signal by applying the GFP booster. And you can see here on the image the difference in terms of quantitation. It's about twice as bright. But also the small size matters. It's not always that tall size or big sizes matters. I think the smaller the better. And that is very, very impressive to see by this publication of the Kaplan lab, or no, it's the Evers lab at the ETH in Zurich at the time in Nature Protocols 8 years ago. If you have, let's say, a target, a GFP fusion protein that you're interested in, you detect it with a primary antibody, you detect the primary antibody with a secondary antibody that is fluorescently labeled. You have a very long, what's called epitope to label displacement distance, right? So here's your target, and that's the fluorophore that you actually detect. Using a nanobody against GFP, that distortion is very small. So here is your target, here is your nanobody, very slow, and here's your dye. So the dye is much closer to the target you're looking at. So this localization error has been examined by the analysis of microtubules that were stained with a GFP booster and conventional antibodies. And what's seen here is the microtubules detected with a classical secondary antibody, and the dimension of the tubules is about 45 nanometers. When you use the GFP booster for the detection, the dimension was about 27 nanometers, and it is known that the size of microtubules is about 25 nanometers. You can see using the GFP booster, you get a much preciser measurement of what's actually going on in the cell. Two images to share these. So this is actin labeled actin in super resolution microscopy. That bar is about 10 micrometers in a HeLa cell, and this is lumentine in a HeLa cell. And here the resolution is even more impressive. It's 2 micrometers. And so these nano labels are really, these nanobodies are really excellent tools for super resolution microscopy. The size of the nanobodies is also very important and beneficial for tissue penetration because they faster and better penetrate into tissue. This is customer data from a transgenic mouse expressing an EGFP fusion protein. Here is a tissue and a primary anti-GFP primary conjugated with fluorophores is applied here, and the diffusion into the tissue is very slow and very limited, whereas the GFP booster equally diffuses into the tissue. So that is very beneficial, and that was a preparation work of this experiment that you can see here, which is a depleted mouse carrying this EGFP construct, a transgenic mouse. And here you can see in green the nervous system of that adult mouse. And this technology is called panoptic V-disco imaging. So here you have all the neuronal connectivity of that mouse, quite impressive. Obviously, you have to deplete or pre-clear the mouse in order to be able to detect all these signals. The stability of the nanobody makes this approach possible. So that's how the neuronal connectivity within a mouse looks like. Yeah, every time I watch this video, I find it amazing. Yeah, so the data is created and done by the Ali Ertug lab in Munich. He moved on from the LMU to the Helmholtz Center in Munich. And this is based on our GFP booster. Yeah, there is the mouse in its total. What I like to show from a different publication from a lab in Shanghai, this is the diffusion time into a 500 micrometer tissue section using IgGs. It will take in between 9 and 11 days for a full diffusion. If you use a Fab2 fragment, it will take about 4 to 5 days. A single Fab fragment, it will take about 2 days. And a nanobody diffuses into a 500 micrometer tissue section within a single day. So the size really impacts the diffusion rate significantly. My last topic is about so-called chromobodies, which are fluorescent nanoprobes for real-time and live cell imaging of intracellular endogenous proteins. And what's a chromobody? A chromobody is basically a DNA plasmid that is visualized upon transfection. It is a non-invasive labeling of endogenous proteins and cellular structures. It does not interfere with the target function nor its localization. That is due to the high binding rate and dissociation rate of the chromobody. So you will always have binding and dissociation, and hence no or very limited interference with the target. It's a very fast no-wash assay. You do not need to overexpress your protein of interest. It has no cytotoxicity or artificial effects, and it's available for actin, lamin, histones, vimentin, and other cellular targets. How does it work? So you have your cells. You transfect your cells with the chromobody plasmid that carries the nanobody and the GFP. The nanobody GFP fusion is expressed, and it will bind, in this case, to the lamina, and you can detect the bound nanobody. And how does it look like? You can do little movies from the first moment here on cell cycle. That's PCNA as the target for the chromobody, on lamin, on actin, DMNT1, as well as on PARP1, a DNA repair enzyme. And what you have to keep in mind is these are structural targets, because the GFP signal will be detectable whether the nanobody has bound its target or not. So a soluble target would not be applicable because it will be very difficult to differentiate in between a bound and an unbound target. Some data from a customer that has detected actin in a zebrafish embryo. That's how a zebrafish embryo looks like. First, I'd like to draw your attention to the head of the zebrafish, and here you can see how the zebrafish is developing. It's a confocal microscopy movie, and the signal disappears when it's running out of the confocal plane. And here I like to draw your attention to the tail of the developing zebrafish, and here you can see how the tail grows into the other direction, as you can see, and also here the signal disappears when it's moving out of the focal plane. So also quite impressive data where you can really follow live in real time the development of your sample. I'm almost at the end of my presentation. If you're interested in learning more about our products, you may visit ptglab.com with all these easy to find sections on our website. I like to conclude. Thank you for your attention. Happy to take any questions, and as always, some disclaimer on trademarks. So happy to take your questions. Yes, please.
So one question regarding the labeling and also the secondary antibody. So what's the difference between these two? Because both of them need to use the primary antibody for the recognition of the target proteins. And for the labeling one, you say you can label it on the heavy chain, the Fc region of the antibody. And the other one, you use another nanobody to do the conjugation. So what's the disadvantages of using these two proteins in candidates?
Yeah, so the question is, what's the difference between the nanosecondary labeling and the flexible labeling, the antibody labeling? I think it's a very fair question. You may consider the antibody labeling as an alternative to secondary antibody labeling. You're absolutely right. In principle, there is no difference. The point on the flexible labeling is it has very, very small dissociation rates. So the affinity is very high. You don't run into dissociation from the primary antibody. That's the advantage. The disadvantage is it carries only 2 fluorophores, so it may not be as bright. For the nanosecondaries, you have some dissociation, not a lot, but it is measurable. However, due to the mixture that is applied, you have a high degree of labeling and it is brighter. Also, the nanobodies are smaller, which is a benefit for super resolution microscopy. The flex linker used in the flexible kit is larger than the nanobodies are. So, but yes, you're absolutely right. You may consider the labeling kit as an alternative to a secondary labeling. Yes, well spotted. Thank you.
Klaus, please open the microphone.
Oh, sorry. Thank you. Yes, please.
So may I understand more about your nano boost and the regular nano secondary? Because you mentioned that the boost one is maybe even better for the tissue penetration. So what's the difference? I kind of confused about the structure and why the booster is better for the penetration instead of the secondary.
Also a very good question. The nano booster is like a labeled primary nanobody. So you only apply the nano booster to the section and you have a 15 kilodaltons protein that has to diffuse very quickly and very deeply and very homogeneously into your sample. If you apply the nanosecondary, you have a mixture of two nanobodies detecting the primary antibody. So you have the 150 kilodaltons, 160 kilodaltons primary antibody, plus 4 nanobodies, a total of 60 kilodaltons. So you end up with like 120 kilodaltons that will diffuse slower into the tissue, still much better than a conventional primary detected with a conventional secondary, but possibly less good than a fluorescently labeled primary antibody. But also well spotted. The benefit is really that you have to consider a GFP booster as a primary labeled nanobody against GFP, and it's the smallest antibody that you can apply to a tissue, and hence you have the best diffusion rates into the tissue. Well spotted, thank you. Yes, please.
So for the chromobody probes, so if I'm like I have the interest in examining some very unique protein, so how do you recognize the specific epitope inside the cell? Because you need to do the construct.
You would need to do the construct, right? So you first need to have a nanobody. The nanobody needs to be suitable in terms of affinity, which is very difficult to adjust because inside of the cell there are different conditions than in a buffer. And so the development of a chromobody is very tedious and very, very long-term project. Also, as I mentioned, you have the signal of the fluorescent protein all the time, independent whether the nanobody is bound or dissociated from its target. So you always have a background. So that's why we stick to structural targets, really. So if it's a soluble protein, you won't see a difference. Our scientific founder, Professor Rothbauer, he claims he has found a method how to differentiate, but I don't get it. It's beyond my head. It's very complicated.
So what if the construct does not work?
The construct, in the literature, you will find nanobodies that have been expressed like as intracellular protein traps, where you have a high affinity nanobody expressed in a cell, and it will bind to their target, and it won't dissociate anymore. In this case, you have an interfering probe. It's like a trap, like an intracellular trap. Our chromobodies are designed in a way that they have the least interference with its target. There is data that I haven't shown about the actin chromobody that has been compared with all kinds of actin detection methods, and it has been found the least interfering with the actin. There is some interference, as with all of the probes, but from those tested, it was the least interfering one.
So a following question here.
Mm-hmm.
So you mentioned about the structural protein to detect.
Yes.
And then, so how about the vesicle or like organelle proteins? Is that suitable for that detection too?
Yes. If you, for instance, add a signaling sequence to a leading sequence, you may target mitochondrial actin, for instance, or ER. That's possible. Yes. We have a custom. Yes, we have a customer that has done experiments like this. Yes. So you can indirectly or directly, if you like, detect mitochondria. If you would detect, if you would have an antibody against, let's say, a membrane protein of mitochondria, you may also detect the mitochondria. That would also work. Unfortunately, we don't have that antibody.
Not yet. And how about the, can you generate stable cell line for that?
Yes.
Constant.
Yes. So, for instance, the zebrafish data that I've shown, that's a stable transgenic zebrafish, and we don't have those cell lines. That's customer data.
Okay.
Do you see internalization of your chromobody probes in the vesicle? How about the vesicles then that can perform further memory imagination inside the cell? Does your chromobody also can be encapsulated, and then outside the membrane we can see these GFP is accumulated inside the small?
I think that's possible, but honestly, I don't know.
And also for the membrane protein, we have to consider N-terminal and C-terminal combination. So your chromobody is only targeting one of the sites. So how can we really observe the distribution of these kind of proteins?
That's a very hypothetical question. I would consider the generation of a nanobody against the C or the N-terminal sequence, or let's say loop or part of a membrane protein being very difficult. Nanobodies or heavy chain antibodies tend to bind only three-dimensional targets. So they need a conformation. And generating a nanobody against a flexible, let's say, loop or just string of two-dimensional target is very, very difficult. It's a good question, but I think the nanobody, you will be very, very lucky to generate a nanobody like this. That would also work as a chromobody. So you add already a huge complexity to a huge complexity and end up in a probably impossible to resolve problem. I'm sorry. We do have nanobodies against peptides. The MIC-Tech, for instance, it doesn't work in immunofluorescence, it only works in immunoprecipitation, And here we looked into the structure, and when you have multiple MIC-Tags, like 2 times or 3 times MIC, fused to your protein of interest, the MIC-Tag starts to form an alpha helical structure. And when we look into the binding of a single MIC-Tag protein versus a 2 or 3 MIC-Tag protein, you see a very, very high affinity towards the multiple MIC-Tags because of that structure being formed, whereas a single MIC-Tag protein has 100 times less affinity, which has certain advantages because you can purify, because you can easily apply MIC-peptide for elution from the nanobody, whereas the multiple MIC-Tag protein can't be eluted anymore. For instance, we also try to get a nanobody against FLAG for years. It doesn't work. So you get a very nice immune response in the alpaca animal, but unfortunately it's on the conventional antibody side, so on the IgGs, but not on the IgGs that are heavy chain only antibodies. So the lecture books say it's an alternative mode of binding, mode of action, and it's supposed to be 3-dimensional structures only. And Flex seems to be a true flexible structure that can be used for immunization. So a nanobody, I think, is good when you have a rigid 3-dimensional conformation. But sometimes you're lucky. We also have a V5 nanobody that unfortunately does also not work in immunofluorescence, however, in immunoprecipitation. And also here we have moderate binding so that you can actually elute. The GFP nanobody has a 1 picomolar affinity. So when it's bound to the protein, it hardly dissociates. So it's not suitable for any kind of purification, but very good for immunofluorescence and pull-down experiments, as it binds and sticks to it. If you want to use it for purification, you have to cleave it. So you need a GFP, let's say a tough cleavage site, and then your protein of interest. So as always in life, beautiful affinities, very high affinities, perfect for imaging, perfect for pull down, but not for immunopresentation. There is no reagent that works for everything. We would love to have one, but unfortunately it isn't. Any more questions? Oh yeah, please.
I have a question about FlexAble body part.
Yes.
I just want to ask any reason for example or from the other producer that this antibody also available to video monitor, not only for the user.
I believe we have customers using it in organoids, yes. I don't think that those data have been published yet. Yes, so FlexAble does work in organoid labeling, but it's not published.
So the same condition also available from the similar for
I would need to ask the customer. I don't know. I would need to ask the customer. But I would think that the labeled antibody is applied to the FlexAble labeled antibody is applied to the tissue. And I guess you may have a slightly increased incubation time compared to a labeled primary antibody, but much faster, much lower incubation time than to a primary that is actually technical for secondary. Yeah, my pleasure. Thank you. I stand still so you don't get in trouble. I've been told I can only move in a certain area over there for our online attendees, but my colleague is very proficient. He follows me. Thank you. Any further question? Doesn't seem to be the case. Well then, thank you very much for your interest, for attending this talk, and also for your very good questions. Thank you very much.
Thank you, Klaus.
Thank you. Let's switch it off.
好，那如果说大家对于这些就是应用上面还有一些问题的话，那就是可以再跟我们贝斯特联络。那我是贝斯特产专 Siri， 所以之后有些产品上的应用问题，不论是Proteintech的抗体或是Proteintech相关的产品应用，那都可以进行跟大家做一个技术咨询的部分。对，那很高兴大家可以来参加我们这次的研讨会。那希望就是之后就是有问题的话，可以跟我们多多讨论，不论是实验上的设计，或者是产品上的应用，或是要解决任何的问题。那再就等等结束之后，我们会有一个午餐，那就是可以就是在外面的时候做一个领取。那谢谢大家。
Can I leave?